All right, so we'll finish up this slide deck. This slide talks about tissue tropism, which you already understand because we talked about it quite a long time ago in this semester. Tropism is a word that's used to describe the spectrum of tissues that can be infected by a virus, and you know that the tropism of some viruses is limited. Viruses that replicate only in a particular subset of types of cells, and other viruses are pantropic, with pan meaning large. They can replicate in many different organs in the body. And when you think about what determines viral tropism, in some cases you know that it is the receptor, and in other cases it is not. And so what this slide illustrates is that you know that ICAM1 is a molecule that is quite ubiquitous, as is sialic acid, as are glycosaminoglycans in the right-hand column here. But the viruses on the left are very specific with respect to the part of the body that they replicate in. So rhinoviruses, we've talked earlier today, replicate in the upper respiratory tract. Influenza viruses in either the upper or lower respiratory tract. Herpes simplex viruses in epithelial cells or neurons. So obviously in these cases, tropism is not determined solely at the level of the receptor. The receptor makes cells susceptible, but not necessarily permissive. So the next slide sort of blows that up a little bit, talks about susceptibility and occasionally receptors playing a major role in tissue tropism, for example, HIV and CD4, but that not being the case, for example, for influenza virus, which interacts with sialic acid. Other cellular proteins can regulate permissivity, even things like transcription factors. So JC virus, which is a papovavirus, replicates in oligodendrocytes because the viral enhancer is active only in this cell type. And there are other examples like that. Down at the bottom, you see information about cell proteases, and we've talked about the fact that cleavage of the influenza virus hemagglutinin by respiratory secreted serine proteases is critical to the pathogenesis of normal variants of influenza virus, and it's only the highly pathogenic viruses, highly pathogenic avian influenza viruses that have highly basic amino acids right at the furin cleavage site allows that HA to be cleaved during assembly. Interestingly, the 1918 strain of influenza virus, which was obviously clearly pathogenic, very highly pathogenic, didn't have these changes. So let's talk a little bit now about viral spread. After replication at the site of entry, viruses can either remain localized or and be contained within the tissue that they infect, or they may spread beyond the primary site. And in that case, we talk about viruses disseminating. And so, for example, measles virus replicates, the primary site of replication is in the respiratory tract, but then the virus disseminates, and what you see ultimately in the skin lesions, the rash, is the product of a systemic infection. And for that to happen, physical and immune barriers have to be breached. So there are some viruses that might replicate in the epithelium and stay very localized and spread back through the respiratory tract. Measles virus spreads from the respiratory tract, ultimately infecting multiple organs. When we think about whether viruses disseminate systemically or are released locally, think about what you know already from our discussions of viral assembly determines that, whether viruses are assembling at the apical surface of the cell or whether they're assembling at the basolateral surface of the cell. And there's information on that in the slide deck that we used when we talked about viral assembly and trafficking. So apical release from a GI surface or a respiratory surface facilitates the dispersal of a virus like poliovirus, which is a GI type virus. The virus doesn't usually invade underlying tissues. Influenza viruses typically are released apically, whereas basolateral release provides access to those underlying tissues and facilitates systemic spread. And here's an example in panel C here of VSV, which is a rhabdovirus like rabies, disseminating basolaterally, being released basolaterally. And I think I mentioned when we were talking about this initially, for Sendai virus, normally apical release allows that virus to be released into the respiratory tract, but one can engineer a mutant virus that can be released both apically and basolaterally, and that can cause disseminated infection. So viruses that produce disseminated infection often do that by entering the bloodstream. And once in the blood, the virus has access to virtually every tissue in the organism. Viruses can enter blood through capillaries by replicating in endothelial cells or through a vector bite like a mosquito bite. Virus in extracellular fluids can be taken up by the lymphatic system, which is very permeable, and then spread to blood. And when viruses are present in the blood, that's referred to as viremia. This slide shows a great summary. It's worth spending some time walking through this yourselves, of the role of viremia and spread in pathogenesis and transmission of viruses. So it really talks a lot about how viruses might infect at the surface, skin, mucous membrane, respiratory tract, GI tract, and then where there is shedding. So for example, herpes simplex virus and papillomaviruses infect here and are shed through that site as well. But then dissemination through the bloodstream can take other viruses to sites like muscle, liver, spleen, blood vessels. There might be replication in some of those sites, amplifying the amount of virus. Then there is something called secondary viremia. More virus, higher virus load in the blood. Some viruses are then shed, hepatitis B, poliovirus, HIV, but as the viral load increases as a result of secondary viremia, viruses may have access to even more sites within the body, and that's what you're seeing here. And then the bottom area of this slide shows where, once there is secondary viremia and replication in distant target organs, where there can be shedding and transmission to other hosts. For example, skin, mousepox, and varicella zoster virus. Those make rashes, rhinoviruses, enteroviruses from mucous membranes, various viruses that replicate ultimately in the lung when the load is high, rotaviruses and reoviruses in the GI tract, and certain other viruses when the load gets high, when the immune system isn't controlling the infection from the can reach the brain. And then this is a slide that we've seen before that basically shows the role of viremia and the sort of route from from the bloodstream to different organs in the host for mousepox. So what you see as a skin lesion doesn't tell the whole story of the series of unfortunate events. When we think about virus in the bloodstream, it can either be associated with cells or virus can be transmitted free in the plasma. And so different viruses use different mechanisms. HIV obviously can be cell associated. You understand that because these cells can be targets for the virus. But other viruses like poliovirus, yellow fever virus, and hepatitis B virus cause viremia, and the virus remains free in the plasma. It's not cell associated. We did talk about neural spread during class today, and for some viruses like rabies and herpes simplex and varicella zoster, neural spread is what you think of. It's the definitive characteristic of pathogenesis. Whereas for other viruses like poliovirus, which is an enteric virus, invasion of the central nervous system is an infrequent sequelae from normal replication and hematogenous spread. And as I said in class today, rabies virus travels from the periphery towards the central nervous system at a rate of 50 to 100 millimeters a day. So, sort of reasonably slow. So better to be bit on the toes than to be bit on the nose. For those viruses that are transmitted through the body, that spread through the body through nerves, this is a good set of tables and an image that describes what that might look like. This slide talks about how poliovirus is typically a fecal oral spreading virus. It enters the GI tract. It binds to those interesting M cells that transcytose the virus. These M cells overlie Peyer's patches, which are little immune organs in the intestinal tract. And then poliovirus typically replicates in lymphoid cells, leading to primary viremia and infection of secondary sites. And again, that secondary viremia, once it reaches a high enough level in the absence of good immune control, the virus can initiate CNS infection. And it can do this either by crossing the blood-brain barrier or infecting neurons, or it can spread by blood to peripheral nerves and then enter the CNS. But typically, antibody, which is an adaptive immune response, limits access to the CNS and prevents paralytic poliovirus disease. These next few slides talk about viral shedding and transmission. And again, viruses are only successful if serial infections can be maintained in host populations. And as I talked about when we looked at the next slide, release can either be from the primary site in a localized infection or from many different sites in systemic infections. And that's this slide again, which is really worth taking a look at. How do viruses get transmitted? Often respiratory viruses in aerosols, 20,000 droplets per sneeze, and droplets of certain sizes are able to reach different parts of the respiratory tract. Fecal oral transmission is more common now in underdeveloped countries. Vector bites are important here to spread arthropod-borne viruses. Healthcare workers are often exposed to blood, and urine is important as a mode of transmission for viruses like hantaviruses. Semen is important as a mode of transmission for hepatitis B, herpes simplex, and HIV, and skin, obviously, for herpes simplex virus, pox viruses, and papilloma viruses. And when you think about things, enveloped viruses tend to be more fragile. We talked about that sort of back at the beginning of this lecture. They tend to be sensitive to acidic pH, and they're often transmitted by aerosols, which are going to be moist, by secretions. There are viruses, enveloped viruses, that are only transmitted really through cell-cell contact. Non-enveloped viruses like poliovirus, a lot of the enteric viruses, reovirus, rotavirus, can withstand drying. They can withstand harsh agents like detergents, low pH, higher temperatures. And these are often transmitted also by the respiratory route or the fecal-oral route. And these are the kinds of viruses, non-enveloped viruses, tend to be the ones that are more stable in the environment. And when you think about, sort of touching something, a doorknob, an elevator button, the toilet seat, think about the cruise ship virus, noroviruses. Those are non-enveloped viruses that are incredibly stable in the environment. So transmission depends upon viral load, where the virus is being released from, the structure of the virus, and the environment. And so the last slide in this deck talks about is box 110 from your textbook, and really talks about some of the urban legends around infections, many of which have to do with infections in the host and populations. And so I encourage you to read this and then be able to call fact or fiction or alternative facts on some of these legends that you might hear.